information contained in this video and/or audio is provided as is for educational and informational purposes only and should not serve as the basis for any trading or investing decisions. Little Grapevine makes no representations and disclaims all express, implied, and statutory warranties of any kind to any viewer or other third party. Neither Little Grapevine nor any of its affiliates makes any endorsement of any particular company, security, product, or financial strategy, and nothing contained in this video and/or audio should be construed as investment advice. Please be aware that we and participants viewing presentations and/or live webcasts and engaging in Q&As may own, buy, or sell shares of featured companies. Investors should undertake their own due diligence and carefully evaluate companies before investing. Please see safe harbor statements of companies participating in Little Grapevine events by viewing their videos and slide presentations at littlegrapevine.com.
Hi, this is Maj Soueidan. I was invited to interview Anthony Cataldo here with GT Biopharma, and this is really an honor to do that. I love doing these things. So we're here with Anthony. We're here with Danny, who works with Anthony on the IR. We're here with us, and I have Bill Langbein joining me, who runs a newsletter called SanaCurrents, which is a biotech newsletter, and he'll get into that in a second. So I just wanted to have a nice little fireside chat with Anthony. Anthony presented at the Little Grapevine a few months ago, and we kind of wanted to get an update where the company is right now with regard to their cancer treatment potential drugs here, and just move on from there. There's a lot to talk about. There's a lot going on in space. The biotech and the nanocap universe has been very volatile. So let's just get right into it, Anthony. So thank you for being here, Anthony. Why don't you just give us the short elevator pitch again, what you guys do again, and we'll get into the conversation.
Yeah, thanks for having me again. For those of you who didn't hear the first one, we're a biotech company, all right? And our specialty and our focus is really on the oncology side. We have a technology called TRIKE, which stands for Trispecific NK Engager. That technology is everyone's familiar with targeted immunotherapy as a buzz phrase. It's really more specifically is, once you really understand the nuance of immunotherapy, it's really more target-directed immunotherapy, all right? Because it really gets into the specifics of what targeted immunotherapy is. There has to be specificity to it. And so our technology came out of the University of Minnesota. Dr. Jeffrey Miller, who is, not arguably he is, the #1 clinician in the NK cell world, whether it's NK cell therapy or NK cell engagers, anything related. He's the most published NK cell doctor ever, and he has created some really astounding technology over the years, made some very amazing discoveries. A thing called Atom 17, which is the reason why clipping, something scientists would understand. There's a clipping of CD16 on NK cells, and I would have to go back to very basics before any of your audience who weren't oncologists or hematologists or biotech savvy people would understand what that is. What that means is when you're taking an NK cell and you're target directing it to kill a cancer cell, one of the problems with that everybody has, except for the TRIKE, is that NK cells, the target for an NK cell is CD16, a CD receptor, which gives off a signal, and it's been noted, its nomenclature is CD16. Everybody uses it to target direct. And that ability to take that into and kill a cancer cell is important, but there's a problem called clipping or shedding, because an NK cell tries to protect itself from being tied into anything else as well. That's human anatomy universally for the beginning of mankind. Even viruses have immune systems. Everything tries to protect itself. And by doing that, it can't be leveraged as much, but we found a way, because of Jeff Miller's discovery, to not have a problem with clipping or shedding, which is why we can separate ourselves. The basics of what we do compared to everybody else. Everybody's trying to get the field of oncology is expanding, and we're getting more and more knowledge of the genome. The trike actually is an entire DNA sequence that would be 4 pages long, right? But at the end of the day, the end goal is to try to get, everyone's familiar with T cells and B cells, which are created in the adaptive side of your immune system. And they've been familiar for a while. NK cells have really started to become more of the forefront now because they are the most prolific cancer killers that the immune system creates, but they've had a number of issues with them that companies like us and our peer group are solving certain issues in them, some more so than others, and causing NK cells to be more leverageable. And an NK, I've always said this, an NK cells were like the general in the immune system, and T cells are more like the soldiers. NK cells can be target directed in ways and can be leveraged in ways T cells can't. NK cells also don't transition into what's called a cytokine, which is dangerous, where T cells do when they become distressed. So there's a lot of advantages to NK cells. The disadvantages until recently is that NK cells only last about 3 hours in your system, so you're constantly having to replenish them. And NK cells also kind of are, in the way everybody else uses them, is a one and done. An NK cell targets a cancer cell, it kills that cancer cell, and then it can't be used again, it's exhausted. And so you got to bring another NK cell in to kill the next cancer cell, whether cancer cells are identified by CD receptors. Also, at the end of the day, what the public would understand is there is acute myeloid leukemia, MDS, throat cancer, breast cancer, that goes on and on. All of them have identifying markers to them. And when technologies, the advancement of NK cells have been really predicated upon the work that Dr. Miller has been in the forefront. His first generation technology is an NK cell therapy, which is actually owned by Fate Therapeutics, which is the largest market cap in our peer field. They're at about a $7 billion market cap. And it's an NK cell therapy. And then there's other NK cell therapies. There's other Nkarta out there. There's Affimed, Dragonfly, Innate Pharma in the NK engager community, which is what we are. We're an NK cell engager. NK cell therapies aren't drugs. They're therapeutic regimens, which include many different things, including drugs and genetic engineering, ablating, which means wiping out the immune system to clear a pathway to generate NK cells, to manufacturing NK cells ex vivo, that means outside of the body, to supplement these technologies that are all out there. So what I'll do is I'll focus on what makes us different, and then we'll go into the data, all right? What makes us different is that everybody's trying to get an NK cell target directed to kill a cancer cell. And to that degree, and just keep it right there, to that degree, we're all successful. We can get an NK cell to kill a cancer cell. When it kills that cancer cell, all the other companies have to bring in another NK cell to kill the next cancer cell. I'll get into us in a moment. And because of that, and the thing called clipping, which I alluded to a little bit earlier in the conversation, they have to go outside and create NK cells ex vivo outside of the body to supplement their products. Or they use combination drug therapies because they can't quite get there in the way that can really do damage to the cancer that we're treating or they're treating. Whenever you do outside manufacturing, that's really expensive. There's genetic engineering involved. They all have to prepare the immune system to accept these technologies because there's, the immune system is polluted with other things, and it's an expensive process. When I started Iovance, Iovance is still a cell therapy company. I always tell folks the most expensive thing you can do in biotech is a cell therapy company. It's $1 million to $2 million per patient cost. And it's really, the ergonomics of it are also difficult. It's highly complicated to administer by the physicians, and the patient experience is really tough. That thing when you hear that sometimes the treatment can be worse than the disease, that's where things like this come from. Chemotherapy is notorious for that radiation therapy, you name it. So all of them are having success doing that. And then there's extra costs. So when I left Iovance, it was with the idea that I love cell therapy. The concept is there, but the cost, the economic model, I think is just oppressive, and you can only service 4,000 or 5,000 patients. And there are millions of patients that need cancer therapeutics because of its cost and its ergonomics as well. I felt that if you could get something that looked like a cell therapy, but was a drug, that would solve the economic sides. Drugs are so much easier to produce and manufacture. And the trike is a small protein biologic, it's a drug. And that's what NK cell engagers are, they're drugs. But ours, unlike our partners, they start off with a drug, they're bispecific, we're trispecific, I'll get into what that means. But they try to leverage the immune system by using all this supplemental stuff, like I said, outside manufacturing, combination drug therapies, and now their products and their cost per patient are back up there to $1 million to $2 million a patient, whether it's cell therapy or not. Another thing that I'll point to, and then I'll say why we're different and what's causing us to be different, is that all of them are spending $300,000 to $1 million per patient in their clinical trials. We spend $30,000 a patient. And it's because of this construct that you're looking at. It's a small molecule drug. It does things different. And it is because of all of those additional things that I said, the issues of clipping and shedding, and then it goes on and on. The trike is a small protein biologic. For those of you who don't understand this kind of technology, if a molecule were the size of the planet Earth, a small protein biologic is the size of a satellite. In biology, small, especially in power, when it has power, can be really powerful. It can get you to places you can't go, the blood-brain barrier, the reservoir where HIV resides. It's a lot of things. So small proteins can be really powerful. This is very powerful. What separates us is everybody else is doing what I said. In the middle of our construct, everyone's targeting their NK cell at CD16. And then the cancer cell binder is the target. The cancer cells for acute binder for acute myeloid leukemia and MDS, myelodysplastic syndrome, which is what our first clinical trial is all about, is CD33 positive. And that's another signal. Nobody can incorporate IL-15 in the middle or use it the way we're using it. Because our patents prohibit that. That's the magic. That's our secret sauce that Jeff Miller created. Interleukin-15 has a symbiotic relationship to NK cells on being able to excite them and cause them to do things differently than ordinarily than anybody else can. NK cells are resident in the innate side of your immune system. And like I said, they have only a 3-hour life cycle. Because of the way Dr. Miller infused IL-15, interleukin-15, in the middle of this small protein biologic, it's keeping IL-15 active at levels never seen before. IL-15 use on its own, everyone uses IL-15, they try to, but you can only administer it at 2 micrograms per kilogram. Anything above that is dangerous, becomes toxic. We published on patient 9, excuse me, at that level at 150 micrograms per kilogram, 100 micrograms per kilogram. We were at 12 times the lethal dose of IL-15 with no side effects, not even fevers. And that's unusual. And the reason and what separates us is when our NK cell, go to the next slide, Danny, or yeah. Before you started, this is real human data. Those are in the green, those are acute myeloid leukemia cells. The little purple guy, purple bluish guy, is actually an NK cell treated with trike. This is a microwell, this is real-time photography and a special type of MRI video. You see it's looking to kill CD33 leukemia. It's killed one cell. But unlike everybody else, it kills multiple cells. It's a serial killer. And it will continue doing this for months instead of just killing one cell and then exhausting and not being able to kill again or just dying because it's over 3 hours. and it will continue looking for CD33, and it only kills its target. What separates the NK technology that we have, the trike, and what our patents are all about, is this. It continues to kill as a serial killer. It persists for months. It also causes NK cells to proliferate. We do not have to go and do outside manufacturing of NK cells to supplement the trike. We don't need combination drug therapies. Everything the trike does is inside the body because of what the trike drug does with its incorporation of interleukin-15, which nobody else can do. When they use interleukin-15 systematically, they're not able to, they can only use it at 2 micrograms, and it can't do what it does in the trike because it's not harnessed in this protective bubble that the trike creates. Those are significant biological differences. The trike is what's called a first-in-class, first-in-human drug. It's never been done before. The FDA, so when we put this trike together and we submitted our data, our animal or preclinical data to the FDA, what the FDA wants to see is they want to see you kill the animals at what level of your drug, the highest dose, is it going to start killing the animals? Because they want to be able to extrapolate that into what the human experience is going to be able to do before they let you put it into human trials. Even at 1,000 micrograms per kilogram, we couldn't kill the rabbits or the mice, but we were killing all the cancer. So they were very confused. They said, look, when is this going to top out? It doesn't work that way. So they understood that, and they allowed us to start our clinical trials. They were very, very conservative with us with our clinical trials because of IL-15, because they know IL-15 at the levels we were proposing would be really dangerous. But they were convinced that we might have something that maybe had harnessed that danger. So our first 2 patients started out at 5 micrograms per kilogram. They said, and if you get past 5 micrograms, that's a very low dose, barely measurable. They said, if you get past 5 micrograms, we'll let you go to the next 2 patients at 10. Same thing. If that's safe, we'll let you go to 25 micrograms with the next 2, 50 with the next 2, 100 with the next 2, 150 with the next 2, and 200 with the final 2. We have now dosed all the way through patient 12 and are looking to dose at patient number 13 at 200 micrograms per kilogram. So let's go to the next slide. What's been our experience on this? This is really amazing. This is we got approved for a phase 1/phase 2 clinical trial. In phase 1, what the FDA wants, all you're looking for is how safe is the drug going to be and what is going to be the maximum tolerated dose level you can use with your drug on human patients. In the first 2 patients, patients 1 and 2, when we bring the patient in, we test many things, but the main things we're testing are how many NK cells do they have in their immune system, and what is their bone marrow blast levels. Bone marrow blast levels are the amount of leukemia or cancer that you have in your bone marrow. It's in the bone marrow that is the most meaningful, not the blood level. Bone marrow is where blood cancers start, like leukemia and MDS. And these patients in patient 1 and 2 at 5 micrograms, they had barely any NK cell levels, and their bone blast levels, their leukemia levels, were expanding by 20% every day. The patients we treat are stage 4 patients with 6 weeks to a couple months to live, refractory to everything else, and they failed 3 other drug therapeutics. They have all been written off. That's the FDA makes you treat the worst of the worst patients when you first start out these kind of trials. So when we tested those levels, leukemia levels, when we tested patients 1 and 2 at 5 micrograms, their leukemia levels stabilized. It didn't progress anymore. That's a win. Even in phase 1 at such a low dose, we didn't expect that. That's important. And what was also important were 2 other things. Their NK cell levels went back, increased, which gave them energy and things that they hadn't experienced before in other therapeutics. And there were no side effects, no fevers, No cardiac events are common with leukemia patients. No cardiac events, no cytokine release storm, which is toxicity, which everybody has problems with CRS. None of that. Then the FDA said, okay, go to 3 and 4 at 10 micrograms. Same result. We tested their levels, NK cell levels went from almost non-existent back to normal, and their disease stabilized. We're seeing stabilized disease even at 10 micrograms per kilogram in very low doses. When we got to patient number 5, it actually kicked up a notch. Instead of just stabilizing the disease in the bone marrow blast levels, remember that's the leukemia levels, we eliminated 33.3% of the leukemia levels in the bone marrow. That is a response. In phase 1, you don't see signals of efficacy. Remember, phase 1's for safety and establishing MTD so you can get to phase 2 and then try to get to these things. Patient number 6 failed because patient number 6 had liver disease on top of leukemia as well. We're only attacking because we're only programmed in this. We have 22 trikes, and this trike is only programmed to attack leukemia with CD33 positive. So nothing happened with that patient. Patient number 7, remember, these are all written off patients that are going to die. Patient number 7 at 50 micrograms per kilogram, we lowered, this was for MDS, myelodysplastic syndrome, which nothing works for. We lowered her bone marrow blast levels from 12% down to 4.6%. That was a 61.7% reduction in the MDS cancer load. That's big. That is a response. Some would call it a complete response. We hold off calling it that because we like to have durable response tied to complete response. Otherwise, it's BS. All right. You want durability. And so this is still in phase 1. And that's now remember, at 50 micrograms, we're now 7.5 times the lethal dose of IL-15 with still no side effects, nothing. This drug is so easily tolerated. These patients aren't used to that at this level. They've been on a torture ride when they get to stage 4 with other therapeutics. Patient number 8 had 2 different types of cancer, multiple myeloma and acute and MDS. We were able to reduce the MDS levels down, but the multiple myeloma was there, and we're not targeting that. So nothing on 8. When we got to 100 micrograms per kilogram with patient number 9, that's now 12.5 times the dose level of IL-15 inside of our trike. That patient's level went down from 22% leukemia levels, this was MDS level again, down to 8%. That's a 63.6% reduction in cancer blast levels. This is in phase 1. You do not see these kind of things happening in a phase 1 trial with still no side effects. We have since treated patients 10, 11, and 12. I can only tell you right now, I can't tell you what the results of those are because we've been approved to present the phase 1 data at the ESMO conference. That's the big European Society conference for immunology on September 16th. Because of that, and they only picked a few companies to do oral presentations, we're under what's called we're blockaded, right? Not embargo. Thank you. My brain slipped. We're under an embargo, and Bill knows what that means. That means we can only talk about it and release it when we're there, which is good news for everybody, by the way. You get a lot more attention than just sending out a press release when you present these in a major conference. So that's happening. We have talked about publicly that we only started to see fever in patient 12. It was a grade 1 fever with Tylenol. We haven't seen anything else. We have yet to see CRS. And none of these drugs that I pointed out to you can say that. So the drug is performing. Now let's fast forward to, and then I'll open it for questions. Go to the Fate Therapeutics right there. In November of last year, Fate Therapeutics ran a study. Well, the University of Minnesota ran the study using Fate's iPSC NK cell therapy, their product. And you'll see with PC3 cells, this is prostate cancer, all right? And in 24 hours using a PC-3 cell, you could see some degradation of the prostate cancer 24 hours. This is vertical, 48 and 72, but not much. When they used their NK cell therapy, FATES, iPSC NK cells, you see it did some damage. It started in 24 hours, it started doing something. At 48 hours, it probably eliminated about 50% of the cancer. And then at 72 hours, it kind of hung around, maybe eliminating another 5%, like 55%, but it couldn't kill it all. When they took IL-15 separately, which everybody can do, no one can use it like we use it in part of our construct, they had a little bit more success on it. You can see the first 24 hours, some killing, the second 24 hours, some disbursement of the prostate cancer, but they were still, even at 72 hours, unable to eliminate it. Then they took the ISPC, their FATES NK cell therapy, and used our trike for which is targeting B7H3. We have 22 trikes. In the first 24 hours, you already see some serious damage. In 48 hours, you see some real serious damage. And at 72 hours, it eliminated all of the prostate cancer. That's data, right? There is no ambiguity to data. Data is what it is. It's not, and that's why I stay focused on data. And I tell folks with CEOs, you don't have to listen to us. You just have to pay attention to the data. And that means that the trike, not only as a monotherapy, can work all on its own, like we showed you, and we have multiple trikes, but it can also be used to take other people's technologies over the goal line. Affimed, Dragonfly, Fate Therapeutics, and Carta. This is the field we play in. All of these market caps are from $1 billion to $7 billion market cap. Our market cap right now is about 300 million because we uplisted. We just became a NASDAQ company in February. We're kind of new to the game, but our data is kind of kicking everybody's ass. Now, any questions?
Anthony, thank you, man. Really good overview. I want to kind of focus on what you just said there last second about how your therapy can maybe be used with other companies' therapies, right?
Sure.
So let's take a situation where Fate right now, as you know, the stock has come down a lot. Now, Fate is, if I understand this correctly, they're injecting NK cells, infusing into the body. Is that they're making it in a lab and infusing it in the body? Is that right, or am I?
Yeah, they genetically engineer an NK cell, right? In a unique way. They can use IL-15 in its traditional way with that in association to it, and they are injecting, they are infusing. All right, their NK cell there. It's still a cell therapy. It's $1 million, all that stuff. They're the best of breed as far as cell therapy is concerned. All right, they're a good company, but they again still have to use outside manufactured NK cells, genetic engineering, and all those things I talked about at the beginning, and the cost levels are up. And so having this data was produced without us knowing it. Actually, we didn't know we were part of that. That was published data, by the way, presented at the SITC conference last year in November.
Who published it? I mean, who presented it?
It was presented by Dr. Miller. Dr. Miller was part of that study.
Oh, okay.
Right. And he also, works with Fate Therapeutics. And this was at a Fate presentation at that conference. All right.
Okay.
And so this is published data. So yeah, we can be an adjunct to not just them, but a lot of other technologies. We get approached all the time now for people wanting to leverage, our targeting modality is very unique, and we're the only ones. Actually, there'll be a press release coming out about our patents next week. Okay, that's all I can say. So we're kind of off and running.
When you look at Fate now, they had some, I think, data a couple of weeks ago that the market didn't like, and I think what you've talked about in the past, when you looked at Fate and their market cap versus yours, you're a little further along in data than they are. Is that correct?
No, not really. They have several products. We're getting different results with an NK engager, which is obviously an off-the-shelf technology that is a lot more economical. Like if our technology continues to progress the way it is, instead of $1 million to $2 million per patient, it'll be 150,000 to 200, and it'll be covered by insurance. Our clinical trial right now, remember Fate and all these companies are spending $300,000 to $1 million per patient in their clinical trials, and we're only spending 30,000. We get 70% insurance coverage right now in our clinical trials for a reason. Okay, so the trikes that we have, what that slide showed for prostate cancer is 20% of the cancer market, the oncology market, is liquid tumors like acute myeloid leukemia, multiple myeloma, non-Hodgkin's lymphoma, etc. 80% of the cancer market is solid tumors. The prostate, that data you just saw, where we were the party that took that technology to 100%, is a solid tumor. Solid tumors are 80% of the market. We have 3 trikes right now lining up, being under manufacturing. At the end of this year, they'll be finished, and we'll start putting them into INDs, getting ready for a clinical trial study, a human clinical trial study with the FDA. We have 16 other trikes here. This is a platform technology, and it's shown several things: efficacy early in a phase 1 trial, and it's shown a very low side effect profile, almost non-existent. And it's shown resilience, persistence, and proliferate. Nobody else can proliferate NK cells in the body. So it's not only a monotherapy, but it can be used as a combination drug therapy, which means we have a big horizon on the head. And so we're progressing. There's a lot of things I can't talk about right now, but we have a big month ahead of us. There's a number of conferences we're presenting at, and we'll be doing that. And we'll see what happens from there.
When you look at Fate's data, Anthony, I mean, I guess they didn't have as an encouraging data release a few weeks ago. When you look at that, right, and Dr. Miller is involved with both companies. I guess I know, is he still involved with Fate to some degree?
Yes, he is.
So when you see that, I mean, is there already a potential discussion about that? Like, would their data have been better if they were using your technology?
Yeah, you're asking me a question I can't answer.
Okay, that's fine. So
Maj?
Yeah.
Can
Go
I
ahead.
follow up on your topic with Fate, and Maj just brought this up about the 7 billion market cap of Fate. And one of the things that that as I was following the GT Bio story and also Fate, is I think that they only had data in one patient for like 3 years. And then most recently they presented data in 3 or 4 more patients. And maybe this is a question the CEO doesn't want to answer, but everybody says their companies are undervalued. So you've got data in 10 patients and they've got 7 billion based on 4 or 5 patients. I do see a disconnect.
Yeah, and I'll tell you what my belief is on that. We were a bulletin board company until February 16th of this year. And as a bulletin board company, nobody's paying attention to us at all. In the meanwhile, we were doing the work. I held off becoming a NASDAQ company until I had clinical trial data. Once we presented the data up through patient 7 at the ASH conference, American Society for Hematology, on December 5th, that's when I went, okay, now I'm going to go up to NASDAQ. And we did it the old-fashioned way. We transitioned from bulletin board. Roth Capital and Dawson James were the bankers that took us forward. I set out to raise 15 million. We actually closed on 28 million that day on February 16th. And since I've had warrants exercised to the tune of we have 40 million in the bank. All right, these were cash warrants at $5.50. All right, but I built an institutional company with no institutional support. Now things are changing dramatically for us. And so everyone's kind of waiting to see what we're going to do next. And we are starting, we went from bulletin board to NASDAQ on February 16th, to the Russell 2000 in June, to the Vanguard and the Chicago Option Exchange over the last 5 months. And we've had a tremendous amount of growth. The stock actually performed, because as I have an analogy that I think addresses your question, Bill. When we were doing the NASDAQ raise, one of the large institutions that didn't come in for the deal, but since has come in on the market, said, asked me the same thing about valuation. And I said, well, let's look at Nkarta. All right. Because Fate's been out there for a while. Nkarta's what, $1.5 billion to $2 billion market cap. They treated one patient in November with no results. Right?
Right.
Yet their market cap is about $1.5 billion. And they said, well, but Nkarta's got, pedigree. I went, well, that's bullshit. Because the #1 person in the NK cell world, by everybody, including you, Mr. Institution, is Dr. Jeff Miller, right? They go, well, yeah. Well, he's got pedigree, right? And they go, well, I created a biotech company called Iovance that has got up to $7 billion in market cap. Yeah, you got pedigree, Tony. I go, so it's not pedigree. The difference is that Nkarta came out with Morgan Stanley and Goldman Sachs, all right, out of the gate. We were a bulletin board company. So we're just now getting the institutions now, and we're getting analyst coverage. We have 3 analyst reports right now, B. Riley, H.C. Wainwright, and Roth Capital, all showing about a $25, $26 price target, which would put us at about $932 million market cap. And there's more analyst coverage coming from larger companies over the next few weeks to months. So we're getting that kind of attention now, but we had to kind of prove it. And to me, I've always said, let the data speak, not the CEO, right?
Well, that's a big raison d'etre for what I try to do, is to let the data speak, and data and events. And that's what we look at and try to capture as you go along. And so I learned of the GT Bio story through Maj, and looked at it, and we wrote a report on it and all the, one thing that, I saw that you had hired a clinical development person in the last month or so.
Yeah, because we're NASDAQ now, we're building our team up. We brought Dr. Greg Burke in. He was with Cellularity as Chief Medical Officer. He's our CMO. He's also President of Research and Development. Dr. Jeff Miller is our Consulting Chief Science Officer. We brought in, actually, I'm going to be announcing some stuff, so I won't tell you. We brought in we're building up our staff daily. Things are happening pretty quickly with us right now.
Well, what I saw with that is, what is the feedback from the FDA in terms of your discussions and planning about when you could begin a phase 2 trial, or the phase 2 component of the existing trial?
Yeah, technically we're already approved phase 1, phase 2, but there are discussions going on that I can't talk about. That I like the discussions a lot.
Is there anything, on the timetable? Is it bigger than a bread box? Is it, shorter than 5 years or something like that?
Yeah, it's there are many things happening in a very short period of time, and a medium period and a more long-term period as well. Remember, we have 22 trikes, and it is public knowledge that we do have incoming from big pharma. Fate, if you look at Nkarta, not Nkarta, Innate Pharma, Affimed, and Dragonfly, they all have big pharma partnerships. So I suspect we'll be in that arena as well. And
Afimed's got some data due, I believe, in December.
yeah, they're a bispecific technology, and
Right.
they work.
Right. Okay, very good. Thanks, Maj.
Yes. And on that, Anthony, when is the actual date of the conference you'll be presenting at?
It's at the conference is September 16th.
Okay, great. Are those things recorded so we can watch them, or is it one of those things you got to be privileged and invited to go there?
I think you have to be part of that, but there'll be coverage of it, right? The analysts will cover it, some newspapers will cover it, that kind of stuff. Here's what I'll say to you and your audience. A phase 1 study is made to show two things: safety data and maximum tolerated dose. Bill knows this very well. I think we're spoiling the market in that we're showing patients that have been written off with efficacy signaling here. We were already a success, even if nothing else happened now, right? You do not see that. I'm not saying nothing else is going to continue to happen, but we're already ahead of the game.
So let's take now you brought that up, let's take a worst case possible. Let's say the data here doesn't show efficacy. That doesn't matter though, as long as that's not really the big. If you have less efficacy in these last 3 patients, it doesn't mean good things aren't still going to happen with the company, right? I mean, this is all about the safety of the dosage, right?
Well, it's safety, but the fact that we're showing efficacy is already meaningful in patients that were written off. Okay. And there's other things, there have been some retreatment of patients that we can't talk about because the FDA protocol, we can't use that data, right? But these patients have had a really good experience with this drug because they're so used to the torture that they go through when something comes in. We had one patient say, "Am I getting saline?" There's no such thing in a cancer trial. Some people think there's blind placebo testing, not in cancer, not in stage 4 cancer trials. That would be unethical. Everybody gets the drug.
Why don't you give us again a situation on your cash, your status now, and I think you talked about that last time, how you had a warrant exercise now too, so you have a lot more cash than then. So talk about your financial position, and how that sets you up for the next few years in terms of your position.
Yeah, we have enough cash to last 2 to 3 years without raising another penny. Now, that's not saying we won't raise additional capital because we have 22 trikes. The more we can put our trikes in our platform technology into the portfolio, the more valuable we become. So we will do an additional financing, but not at this level. I'll wait until we get to that $20 level, and then I'll think about it. But having another $100 million on your balance sheet just attributes. Remember, we only have 31 million shares issued and outstanding. Fully diluted, we're at 33 million shares, and that's another $12 million worth of warrants out there that are exercising not because we force them, because they're making money, because the stock has had biotech sector has taken a real big hit over the last 2 months, particularly the last. And we're part of it.
You see Sava, that got just killed a few days ago again, Bill.
We're now part of the NASDAQ.
Sava?
Yeah, we're now part of the Russell 2000, so we got hit by it too. But nothing's changed with us, all right? We're progressing.
Yes,
when you talked about the cash, in a good scenario, you get to the design of the phase 2 component for your drug in the next few months, and you would fund that yourself, and you have the cash to fund that yourself with what you have on hand.
Yeah, not just that, but 4 other trikes as well, all the way through phase 1 and phase 2, without needing a penny.
Right. And would you do the phase 2 through the University of Minnesota?
No, we're now expanding beyond that. We're going to continue using the Masonic Cancer Clinic as a clinical trial site, but we have several other trial sites ready to go. You're getting ready. One's ready to go right now. There'll be about 8 different clinical trial sites, which will advance the timeline significantly by treating multiple patients in multiple trial sites.
Would it be possible to go to a phase 3 after the review?
Yes.
No, I don't mean, I mean like jump right from the phase 1 to a phase 3.
No, there's no such thing. What you would be talking about then is a phase 2B pivotal study.
All right.
Right. And that's a possibility depending on data and things like that. And that means you could actually get approval at the end of your phase 2B. You would still have to do a phase 3 study, but you would already have your commercial license. And there's things like fast track and orphan drug status, and we'll see how that goes. There, like I said, there's a lot of stuff I can't talk about.
I understand. Thank you.
Cool.
All right, well, thanks, Anthony. I think you covered it well. I mean, you answered a lot of our questions the first time around. We sent you, I think, like 20 of them or so. And but I think thanks for bringing us up to date here. I mean, I guess really the next thing here is we're all waiting for is that September type of conference and see what that data looks like. That'll be pretty big for us all.
Yeah, what I can say is we're now, because everyone's advised me to do such, because no people don't really, they're kind of looking at us like, let's take the analyst coverage we have. That valuation of $25, $20, and $26 price target is based on one trike in that are the current trike going on for AML and MDS in only the US market. And that market's worldwide. We got 21 other trikes and global markets and stuff like that. So when I was asked that question, Bill, by one of the analysts covering us, he said, "What do you think your valuation is?" I said, "Well, isn't that your job?" You know? And I said, and I always go shy because CEOs are constantly saying we're undervalued. His comment to me was, "This is the first time I'm going to say this. You're the most grossly undervalued company I've seen, period."
Yeah.
And I went, well, you know, we're kind of, it's the rabbit and the tortoise race, you know, the rabbit's way ahead.
Right.
So I just feel we're going to win the race because we're steady. And we're very consistent. We let the data speak.
And one last thing on the trike side, I think so you have, you said 16 other trikes in the pipeline? 16?
Yeah, we have 1, 2, 3, 4, 5, 6. Yes.
I'm sorry, 6. I'm sorry, not 16. I'm sorry, 6. And so you've already done some preliminary testing.
Oh yeah.
Those are
presenting more and more trikes over soon as part of this process. We're going to be talking about it in the conference. So we're going to be opening up the platform, that I can say.
Right. Great. Okay, awesome.
Yeah, this one is for ovarian cancer, right?
Right.
7H3 that worked in that prostate slide you saw in the combination therapy.
Yeah.
Also works for ovarian cancer. And if you look at the tumors, these are in the peritoneal cavity. They're nasty tumors on these mice. When you hit them with just IL-15, it does something, but not much. When you hit them with our trike, which includes a different version of IL-15 no one else can use, and the way we use it, you can see it's eliminating the ovarian cancer. So that's just another example. B7H3 is a multi-basket trial where it will hit prostate cancer and ovarian cancer at the same time.
Excellent. Thanks, Anthony. And if I have any more questions, I'll send them along the way, and maybe you can answer those in between now and then, and get those out to the network.
Thank you, Anthony. Thank you, Danny.
Hey, Bill, why don't you give yourself a little bit of plug what your newsletter does really quick, because we really didn't get into that yet.
Okay. It's alternately been described as a newsletter or a sports book for biotech. So, what we do is each week we have we select a catalyst that we kind of cover. So we cover an anticipated catalyst like the ESMO meeting. There's a company that's going to present data there, and I think when the data is revealed, much like will happen with you, the stock's going to rise on the data. And so, generally we want to get a pretty good spread from like 5 to 20, at the start, to maybe 35 to 50. And the idea is that, you know, we make the calls on the catalyst much more so than the company, but then there's other reviews of the company, you know, for the long-term prospects that we provide.
Yeah, I'm okay with catalysts. I think, you know, when you drive based upon, you know, events, you know, milestone events happening, you don't suffer from that gigantic up and down curve, right? You progress based upon execution.
Right. So, you know, that's, you know, that gigantic curve of up and down, you know, there's not a lot of biotech companies that escape it unless it's something like, you know, Fate, which has a Red Mile and many others behind it. So they keep pumping it up. But for most people on the biopharma and medical device stage, it can be an up and down term, you know, on the development innovative side. And
Yeah.
this year, you know, there's been, well, it's just been a bad year for small cap biotech, but it's starting to recover.
You know, it's been a good year for us, right? We were a bulletin board company at the beginning of the year, trading at 20 cents. All right, we're now 870 or something like 850, whatever.
Well, my job was to get you at 20 cents, but I didn't get that anyway.
Yeah, thanks, Bill. Thanks, Anthony. I really enjoyed it.
Thanks, guys. I appreciate it.
Thank you.
All right, bye-bye.